to observe it and you have to manipulate it with the scientific method to show it's a virus. So these pictures that we're going to see a lot of, I'm assuming, as you said here, every single photograph of a virus, you will not, it's from a cell culture, you will not actually find it from the person. And that is the problem. I mean, Mark, episomes, I'm sure he's making fun about exosomes. Yeah. Whether it's apoptotic bodies, microvesicular bodies, exosomes, whatever the heck they want to label them, they're usually a product of a cell culture, or at the very least, just kind of artifacts. And so just pointing and declaring, that's another big thing that virologists do, is that they'll do this whole bastardization of the cell culture process and then take a little picture of a little slide. It's not even a video. You know, they say a virus is going into the cell and replicating. They can't show that. They just take a tiny little picture of something they claim is happening without actual proof of it. It's like taking a picture of a tiny white strand of hair and then saying that's proof of a unicorn. It's the same exact process.
Right. And Gilbert Ling and Harold Hellman, two biologists, and several others as well, have done a really good job breaking down the problems with electron microscopy as it pertains to biological samples. Because ultimately you're looking at a dead, stained, sometimes heated, sometimes dehydrated, static image of something submicroscopic or microscopic, and then claiming you know exactly what that looks like when it is alive. And then the added problem with, I'll get into it in a minute, but I'll just share here just to reemphasize the point. I'll reemphasize it later. The problem with virology specifically is that is coming, all those steps that they take to prepare a sample for electron microscopy, after all of those other insane steps are done, where they take fluids from a sick person, add it to viral transport medium with antibiotics, antimycotics, fetal bovine serum, add those to a foreign cell line that is kept alive in a lab via artificial means, add a bunch of other substances to that. The cells then break down into a bunch of particles, and then they call the resultant particles viruses. Okay, so a negative control is a control group or sample and experiment that is not exposed to the variable X that is being studied. So again, with respect to virology, they've never demonstrated that X is even there in the first place. They say that it is there, but it's assumed to exist in the fluids of a sick person. They've never demonstrated that it's actually in the fluids of a sick person. So they can't conduct an actual control experiment to begin with, because they never have the independent variable. Now, again, if we're making concessions and we'll play with their play on their playing field, so to speak, we could say, okay, you say that the virus is too weak or there's not enough of it in the fluids, so we'll play with that for a minute. Even if we make concessions to that degree, when they do what's called a mock infected culture, they either use different concentration of antibiotics or antimycotics, or at the very least, you could say they don't add fluids from a healthy person, or a combination of both those things. So they're ultimately not treating the quote unquote control, as they claim, in the exact same way as the experimental culture, which is the whole point of doing a negative control. That is the entire point of it, is to treat it exactly the same to see if the thing you think is the cause, and again, in this case, they don't even prove that the virus is there to begin with, is actually the cause, or if it's actually something to do with the other substances and other variables that are present. Okay, so when Dr. Mark Bailey, for example, and he highlights this in his book, in his paper that is available publicly, it's now been turned into a book as well, A Farewell to Virology, has contacted the authors of some of these papers. They admit that they use a lower concentration of antibiotics and antimycotics in the quote unquote mock infected culture. And their reasoning, as I understand it, is because, well, we're not adding human fluids, so we don't need to use as many antimycotics and antibiotics because there's just not enough vectors of bacteria and fungi present anyway, so we don't need to. And you see the baked-in assumption there is that antibiotics and antimycotics are purely beneficial, not bad for cells. So because we don't have a human sample, we don't need to use as many antibiotics and antimycotics. So, Jacob, I don't know if you have anything to add to that, and then I'll get into some of the...
Yeah, I mean, as you said, the mock is supposed to pretty much validate the existence of a virus by saying, oh, see, there's no negative effect to the cell, therefore there's a virus in the other cell culture. The problem is, as Mark stated, and I have the paper here in case, the Farewell to Virology Expert Edition. Get it, guys. It's free. Just print it out and read it. In any case, these mock infections, mock negative controls, we were actually able to find a couple that use entirely different cell lines. They used entirely different concentrations, as Alex said, of antibiotics, antimycotics. And it's not even just lessening the usage of these toxic chemicals like antimicrobials. That's obviously a given. But simply not adding the same sick fluid is a problem, because fluid in itself has a lot of proteins, has a lot of enzymes. It will have obviously microbes. It's going to have food particles, inflammatory waste products, all that stuff. So them not adding that, and not even adding fluid from healthy people, they really don't add anything to these things other than these antimicrobials and maybe some growth stimulants. That's an issue in and of itself. That's not a proper control experiment. And that's why a lot of people, even in our camp, will say that what John Enders did a long time ago wasn't actually a proper control experiment, because he didn't use the healthy fluid either. So these mocks are just a slap in the face of science in general. They're horribly done, because they're not actually having something like an independent variable, obviously. But they don't, when questioned, they literally do not understand why it is what they're doing is not scientific and not a proper control. It's actually insane to me. But yeah, that's pretty much it.
Well said. Okay, and this is where we'll just briefly touch on the Enders so-called measles virus isolation technique. I'm not going to get into all the details because this presentation is already going to be incredibly long. But if you want, you can look it up and find it pretty easily, and you can go read it for yourself. All of the substances that went into his quote-unquote measles viral isolation technique. Here's a list of those substances, and then cytopathic effect was observed afterwards, meaning the cells experienced cell death and decay after all of these substances were involved in the process. Again, throat washings, blood, feces from measles patients. Again, this is a common issue with respect to people just learning about the problems with virology or being opposed to it. And I saw it yesterday. All respect to Dr. Suzanne Humphries, by the way. She's done incredible work with her book, Dissolving Illusions. But she and I got into an exchange in a comment section on a post that was made by the High Wire from my appearance on the High Wire. And she said, something to the effect of, anyone who claims viruses don't exist. Actually, here, let me just bring up the quote, because this is a perfect example.
Yeah, bring the receipts out. Like, let's talk about it.
Yeah, and like, again, all respect to Dr. Susan Humphrey. She's done incredible work. I just don't think she's actually looked into this as much as she claims she has. So this is what she said. It's not black and white, terrain and microbe. If this guy, referring to me, thinks there's absolutely no evidence of viruses, he simply hasn't done enough research. It's a silly thing to say. Yes, there's corruption in virology. Yes, HIV is a construct of sorts. Yes, COVID was laboratory made. We'll get to that. But don't tell me that kids don't catch chickenpox at chickenpox parties and measles at measles parties, and then they just never get it again. So you can't call that a toxin. So that's a straw man of our position. We're not saying everything is just based on toxins. The second thing is, that is not only affirming the consequent, which we'll get into here in a little bit, but it's conflating cause and effect. The very clever, whether it's intentional or not, thing with these conversations is that, for example, when we talk about measles, if you make the statement, the measles virus has never been demonstrated to exist, people take that to mean that we're saying measles, meaning the symptoms don't exist, because measles virus, measles symptoms have the same name. So what we're doing here is we're not asserting either A, that the real symptoms don't occur, nor are we asserting B, that the symptoms, I guess really just A, that the symptoms don't occur. But what we're doing is questioning the cause.
Yeah, we are differentiating the symptomatology from the presupposed cause. And that's
exactly
why we say no one says people don't get rashes when they're diagnosed with whatever measles is. What we're saying is the rashes were not caused by a measles virus. That's
correct.
And the people kind of, you have to formulate that. You have to walk them through that, because they don't distinguish those two.
Yep, exactly. Well said. So in this case, right, so he had people that presented with these rashes, and he was trying to figure out what is the cause. I don't know why he did all these insane steps, but this. I think it kind of is a window, and this is pure speculation here, into when you're determined to figure out the cause and you have a presupposed idea. He had the presupposition that, well, it's not bacteria. It must be a particle that's even smaller than bacteria. You're going to do whatever you can to make the effects in a lab in order to claim victory. And so he did all these steps, and then the cells experienced cytopathic effects. But what's interesting about Ender's quote unquote measles isolation technique, and this is a quote from the paper, is that he did the exact same process, except he did not include any sample from a sick person, meaning he followed this exact same procedure here, throat washing, milk, penicillin, streptomycin, all these substances put on a rhesus monkey kidney cell, but he did not include any possible source of the measles virus, quote unquote. No throat washings, blood, or feces samples from a measles patient. And ultimately, he said a second agent was obtained from an uninoculated culture of monkey kidney cells, meaning it did not include any inoculation from sick people. The cytopathic changes it induced, meaning the cell injury or death, in this unstained preparation could not be distinguished with confidence from the quote unquote viruses isolated from measles. What that is saying in layman's terms is that he had the exact same resultant particles, or very, very similar, let's say, to be more specific, resultant particles following the exact same technique without including any possible source of measles, meaning he has then invalidated the foundational evidence that he came up with. But John Franklin Enders went on to win a Nobel Prize for coming up with the measles vaccine. And I think this was sort of just ushered in for the entire field of virology, completely overlooking this line and a few other lines in the Enders measles isolation paper. So there have been some modern modern versions of this in the COVID era. One by Stefan Lanka, who is himself a former virologist. He would say he thinks that he also is pointing out the lack of validity in the field of virology, having himself been a virologist. So when he did the exact same technique that virologists do, using the exact same concentration of antibiotics and antimycotics in fetal bovine serum that most virology papers use. Sometimes, again, you see here on the screen on column 3, sometimes they use 0% fetal bovine serum. Sometimes they use a different type of antibiotic. Sometimes they use antibiotic and antimycotic. Sometimes they use Earl's minimal central medium rather than minimal nutrient medium. They use varying degrees of this, but in general, he followed the exact same technique that virologists follow, except he did not include any sample from a sick person, meaning no possible source of any quote unquote virus was present. And it resulted in the exact same cytopathic effects that virologists point to as quote evidence for viruses. And again, I just would like to emphasize this because even when we point this out, people don't get this. This is the foundational evidence for the entire field of virology. Yes, they point to electron micrograph images and other things like this. This is a light microscope image that you see here, and we'll get into that in a little bit. But this is the foundational evidence, and it has been invalidated. It has been falsified via a variety of ways. It would be like someone claiming the existence of unicorns, pointing to this hair and hoof prints they found in their lawn, and then someone else saying, well, look, I have a horse ranch right next to my house, and I find hair and hoof prints in my lawn too. That's what we're talking about here. This is the foundational evidence for the entire field of virology. It has been falsified multiple times, by the way. Here we go on another example. So over the course of the last couple years, there's been some biochemists and geneticists that have conducted even more thorough investigation into the cell culture isolation technique, following the exact same ATCC protocols, which are the protocols that virologists follow for their cell culture isolation technique. And they achieve the exact same cytopathic effects that virologists claim as foundational evidence for viruses. Not only that, they used a more robust cell line, according to virologists. So a cell line that is less prone to experiencing cell injury or death, more robust. And they use a less harsh version of antibiotic, penicillin, streptomycin. And not only that, to confirm the occurrence of cytopathic effects, they employed what could be called objective verification methods, laser spectrometry, and count-test cell viability analyzer, meaning they employed a technology rather than analyzing the electron micrograph images or the light microscope images, rather with their naked eye and pointing out the cell injury or death. They used an objective tool that measures the amount of viable cells in a dish. And so here's an example of them using, for example, 10% concentration of fetal bovine serum and only 1 times concentration of penicillin streptomycin with this HEK cell line. So they see using Count-S, 95% of the cells are alive, only 5% of the cells have experienced cytopathic effects. And again, this is an important piece of context from the American Society for Microbiology. The rate of cytopathic effects is also a characteristic that can be used to help identify viruses. Again, the rate of cytopathic appearance is also a characteristic that can be used to help identify viruses. In general, the rule of thumb is that a virus is considered slow CP if CPE appears after 4 to 5 days in cultures inoculated at low MOI, and rapid if CPE appears after 1 to 2 days in cultures inoculated at low MOI. Basically, what this is saying is if the culture, the cells experience cytopathic effect at 4 to 5 days, that is evidence of a virus. Sometimes 1 to 2 days, also evidence of a virus. Basically, what this is pointing to is that CPE is foundational evidence for the existence of viruses. Okay. So here, 32% of the cells have experienced cytopathic effects. Here, 34%. Here, 30%. Here, 35%. Here, 36%. Sorry, something's in the way of my screen right here. Here, 36 again. And this is just by reducing the nutrient medium from 10% down to 2% and then down to 1%. Again, no human sample was used in any of these cultures. So this, again, invalidates, falsifies the foundational, quote unquote, evidence for virology. Not only that, they sent some of these cultures off to an independent lab to conduct electron microscopy on them. And they found images that are morphologically indistinguishable from what virologists refer to as viruses. But again, there is no possible source of viruses present in these cultures. So here on the left is CDC image of SARS-CoV-2. On the right is this team's image of what looks morphologically indistinguishable, same size, same shape, same appearance as SARS-CoV-2, but there is no possible source of SARS-CoV-2 present. Here, HIV, same exact thing, same size, same shape, same morphological appearance. Here, measles, same size, same shape, same morphological appearance. And again, there is no possible source of measles, HIV, or SARS-CoV-2 present in any of these cultures. Jacob, so far, do you have anything to add?
No, I mean, these experiments on top of Lanka's experiments, in my opinion, completely falsified the cell culture stuff just by experimentation alone. In principle, these cell culture experiments are pseudoscientific, so they don't need to be falsified. But it's just good to see actual pictures of these things because it helps people understand that just seeing a picture doesn't mean anything. These little particles, as shown in these experiments, there's no way on God's green earth that these could be measles, these can be COVID, these can be HIV, because there was no trace of any sort of viral particle or viral sample in the medium that was used. These are all just straight up how they do it, cell culture processes, creating the same exact particles. And of course, depending on who you ask, they're gonna label it something different. They're gonna say it's an apoptotic body. They're gonna say it's a microvesicular body. They're gonna say it's a virus if it's a virologist. But in terms of just pointing and declaring, this is how easy it is. You can just point to something, claim it's something, and then use that as a basis for your entire study.
Yep. Well said. Okay. So outside of these procedures that have been done to follow the exact same technique that virologists follow, there's also countless examples of researchers, biologists of various types, finding particles that are morphologically indistinguishable from what virologists refer to as viruses, but by the author's own admissions, they couldn't possibly be, quote, viral. So we have observed morphologically indistinguishable inclusions within podocytes and tubular epithelial cells, both in patients negative for coronavirus disease 2019, as well as in renal biopsies from the pre-COVID-19 era. This is published in Kidney 360. Basically, what they're talking about here is they're finding particles that are morphologically indistinguishable from what virologists refer to as SARS-CoV-2 in patients who are both negative for COVID-19 and also in patients prior to the COVID-19 era in these electron micrograph images. And again, this one is just another example of that. Electron microscope images in figures A through C do not demonstrate coronaviruses. Rather, the structures described as viruses are clathrin-coated vesicles. This one, most of the published images depicting the suspected virus are very similar, if not identical, to multivesicular bodies. This one, I respectfully offer an alternative interpretation of the data. The structures identified as SARS-CoV-2 virions look exactly like clathrin-coated pits or vesicles. So we could give dozens and dozens, if not hundreds of examples of this, and I highly recommend checking out where I have his stuff here, Virology.com, or just check out Jacob's Instagram, Undercover Virologist. Both have done a very good job of breaking down the various electron micrograph images that are claimed to be viruses, or some mini examples where the authors, as I said, share that these could not possibly be viruses, but they look morphologically indistinguishable from what virologists refer to as viruses. So again, not only has the cell culture isolation technique been falsified, also electron micrograph images regarding quote unquote viruses have been thoroughly falsified at this point. Again, finding particles using the same cell culture quote isolation technique that are morphologically indistinguishable from viruses, but no possible source of a virus being present. Okay, so this brings up a point that I also touched on earlier regarding this is coming from Harold Hillman's work. Unfortunately, he's passed away. Cell biology is currently in dire straits. I highly recommend reading that paper and some of the other work that he's put out. Also, some of the work from Gilbert Ling, basically sharing about not only the issues with electron microscopy, but the issues with cell biology as a whole. And just, again, as a larger point, without even getting into the tinkering that they do at the microscopic and submicroscopic level with these cell lines that are artificial, kept alive via artificial means, taking something out of its natural context and bringing it into a laboratory environment, and then trying to make assertions regarding what occurs in nature based on what happens in a lab is already itself problematic. So, as Dr. Harold Hillman says here, when a tissue is prepared for electron microscopy, an animal is killed, the tissue is excised, it is fixed or frozen, it is embedded, sectioned, stained, radiated by quote unquote electron beams. So, if I were to take you in your natural state, any of you listening or watching this right now, and do that exact same thing to you, do you think you would look like you did in your natural state? Especially because in most cases, we're talking about something that is a static image. Do you think it would be appropriate to then make claims about how you walk, talk, move, et cetera, et cetera? I think any of us can answer that question and say absolutely not. That would be absurd. Okay. So the big question that comes up oftentimes, especially during the COVID era, is what about gain of function? I just want you to take in this image if you're watching this right now. I know for me, at the time when I was seeing these images come out, having thought at the time that, viruses were real things and that they had successfully manipulated one in a lab, I was watching this happen from November, December onward. I saw these images come out and the video clips that came out of China, and I was terrified. I'll be honest. And then by the time the quote unquote virus allegedly made landfall in the United States, I saw that what these images were really just propaganda. But think about what this conjures up inside of you, what fear response this leads to. And we'll get into some things related to mass psychogenic illness, mass social contagion, nocebo effect, and psychosomatics here in a little bit. Okay, so this idea that gain of function was censored originally, therefore it must be the true thing. I know that's a very broad frame that I just made there, and I think it almost, yeah, I guess just a broad frame, but this is sentiments that are shared publicly amongst the health and freedom community quite a bit, that they censored gain of function and they're censoring, therefore that must mean that it's has some truth to it. So here's the problem with that though. Prior to censoring the gain of function narrative, which I do acknowledge a lot of social media platforms and just the media in general did do that, I did a custom Google search from roughly December of 2019 through May of 2020. And I just typed in some keywords like gain of function, coronavirus, gain of function, Wuhan, COVID, lab, Wuhan. And I found dozens and dozens and dozens and dozens of mainstream articles openly talking about the idea that COVID came from a lab, that gain of function research is dangerous, that it might have been deliberately released from a lab, or it might have been accidentally leaked from a lab. And ultimately, this idea was seeded amongst the population and then censored. So think about the psychological effect that that might have on people to seed this idea amongst the population with mainstream articles and then censor it, accompanied by images like this and video footage of people dropping dead. Right? Okay. So a lot of people refer to Dr. David Martin, the bow tie guy from the Plandemic documentary series, as evidence. Oh, someone just said this slides are stuck. Can you guys still see? Is everyone seeing this slide with David Martin and me up on screen? Okay.
Yeah. What about gain of function? Yes.
Yeah. Okay. So a lot of people that have bought the gain of function narrative ultimately refer to people like David Martin, who has given testimony in front of various political councils, has given testimony in front of the European Union. He was obviously featured in Plandemic, talking about gain of function research. And they assume that what David Martin is talking about, because in my opinion, he is too vague with his language. They assume that he is talking about a particle or a virus that was weaponized in a lab and then deliberately released amongst the population where it was causing illness. So this is a clip from my episode with David Martin back, I think, 2.5 years ago at this point, where I interviewed him and asked him to clarify his position on whether there is an actual particle made through gain of function. So I'm just going to play this real quick. Let me stop share for a second and make sure I have my volume on. Liev, how are you doing on time, by the way, brother? Okay, great. All right. Real quick, though, to ask a very direct question. Can everyone hear that? Can everyone hear that?
Yes.
Cool. Okay. Rewind it real quick. Real quick, though, to ask a very direct question. When you say that there is a biological weapon,
yes,
and you're now saying that you don't think there is a biological weapon, or do
Nope,
you? I don't know.
I didn't say that. There's
Okay,
absolutely a biological weapon. It is a
Okay, so what-
biologically derived toxin.
Okay. So what is the application of this biological weapon according to you then?
The application is injected into people. It is actually a pro-inflammatory, pro-oncologic trigger to actually harm and kill people.
So the biological weapon is coming in the form of injection.
That's correct.
Okay. So is there
No question.
Okay. I don't disagree with that. Is there a biological weapon that is being spread from person to person?
Never has been.
Never has been. Okay. Oh, I think we can agree on that. Okay. So David, then my question is, given what you just stated there, don't you think it's important to clarify that publicly?
No, because here's the problem. Listen, I went up to Capitol Hill and I sat down with Senator Ron Johnson.
Okay, so after that, he basically went into, and I understand, I even understand his perspective better now, although I disagree. This is the problem. Like, me understanding his perspective doesn't mean that I agree with where he's coming from. I just understand where he's coming from. If what he's saying is his genuine position. He claims that he uses this vague language rather than clarifying that there is no transmissible particle made through gain of function, because he's using their own language against them, trying to pursue prosecution and hold people accountable. My response to that would be, well, where is the holding people accountable? We still don't have any of that at this point. And again, I shared this on the high wire because Dell kind of said the same thing. If there is no evidence of the foundational claimed cause that led to all these things, I mean, I'm absolutely not an expert on, the different ways that people are manipulated to do what is fundamentally harmful to them biologically. But I would imagine that turns it from, Fauci could say plausible deniability because, it accidentally leaked from a lab too. Wait. There's actually nothing at all. There's no evidence of the initial cause. And I'm not saying there was nothing in terms of there wasn't actual symptoms of illness occurring over the last few years. But what I'm saying is there was no evidence of the foundational claimed cause. That turns it from plausible deniability to, oh, shoot, okay, we might have, I don't know, this is a big claim, genocide or something like that on our hands. Like, it makes the situation a little bit more serious and a little bit different. And I also want to emphasize that there have been some court cases either won or dismissed related to the idea that viruses have not been demonstrated to exist. I'm going to get into some published research on quote unquote gain of function here in a second. Before I get there, Liev or Jacob, do you have anything to add to this?
I'll say one thing regarding the gain of function stuff. And you mentioned it. A lot of the critique that we receive on this end is, so you're saying Fauci and all these people don't deserve to be tried for their crimes for making a virus, yada yada yada. No, that's not what we're saying. We're saying they shouldn't be tried for making a virus. But in terms of hospital protocols that killed, thousands and thousands of people, that remdesivir, the use of ventilators, steroids, all these things, the making of a vaccine that is literally the most deadly vaccine in history, the psychological torture they put people through with propaganda and masking and social distancing, what they did to an entire generation of children growing up in that era, they should be tried for that. No one says they shouldn't be held responsible for that. They shouldn't be tried for making a virus or a gain of function, because if anything, that kind of deflects away from their responsibilities on the hospital protocols and the vaccines and everything. Because then it actually gives them like an excuse of, we made the virus, but we tried to fix it with these masks and the vaccines. No, it's actually the other way around. We faked this narrative, destroyed people's lives by perpetuating this virus through killing people in the hospital and a vaccine. So that it's just the flipping of the responsibility there. It's there totally should be held responsible. It's not going to happen. Let's just be real. It's not going to happen right now, at the very least. We see what they're doing right now with the psychological warfare on the Epstein files and all that. We're waiting for something to happen. Nothing's going to happen at this juncture. So even having this conversation on, who should be held responsible, it doesn't matter. It's really on us. Where do we pass that responsibility on? The health is really our responsibility. Let's stop blaming other people. Let's just stop believing what they tell us. That's really the core issue.
Well said.
I think my emphasis on the gain of function stuff is that often it puts us in a greater fear state than just the whole virology narrative in the first place. Like we want to break free from the fear. That's what the terrain paradigm is about, right? And so the gain of function keeps us there. It's not, Alex pointing out very clearly here, there's no empirical basis, there's no logical basis for any of this stuff. There's no evidence that this actually happens. It's just another fear story. And this is the point of all of this, is to break free from the fear, right? It's just it's a way of taking people who woke up and keep them in the same fear mindset, or it's a permitted narrative. So anyways, that's my emphasis.
Well said. It's another example of a false dilemma. We have so many examples of that amongst society right now. Real quick though to ask. Okay, so here's an example of a published gain of function, quote unquote, experiment. In one of her videos, I don't remember exactly which one, Dr. Sam Bailey at drsambailey.com does a really, really good video breakdown of this particular study that was done. but it is titled Airborne Transmission of Influenza A H5N1 Virus Between Ferrets. In the abstract, they describe to address the concerns the virus could acquire this ability under natural conditions, we genetically modified H5N1 virus by site-directed mutagenesis and subsequent serial passages in ferrets. The genetically modified H5N1 virus acquired mutations during passage in ferrets, ultimately becoming airborne transmissible in ferrets. Ferrets, not parrots. Okay. So what was done in this study? So first off, this experiment did not start with purified viral particles, but rather a mixed cell culture that the researchers labeled as H5N1. When they first tried to show airborne spread between ferrets, it did not work. They then began a serial passage process. They basically had these ferrets in cages that had airflow going from one direction in one cage to the other one. So they would inoculate one set of ferrets with the substance that they claimed contained H5N1, and then have air flowing into the ferret's cage that was set right next to it. So they did serial passage. They put the material into a ferret's nose. Again, this basically unpurified mixture claiming to contain H5N1. And then they killed the ferret a few days later. They then mashed up its nasal tissue, put that mixture into another ferret's nose, and repeated this multiple times. And again, what was being quote unquote transferred each time was a crude biological mixture, not purified virus. And as they repeated this process, they detected genetic changes in the samples and interpreted that as the virus mutating. Okay, and then as with respect to their evidence for infection, that relied on cell culture tests, again, using dog kidney cells here, and genetic sequencing, as I just said a second ago, which are indirect lab methods rather than direct demonstration of purified particles that cause disease. After several passages, they repeated the airflow cage experiment and reported that 3 out of the 4 ferrets showed, quote, signs of airborne transmission. That's what they claim. Here's the issue with that. None of the recipient ferrets became critically sick. Researchers collected nasal swabs from the recipient ferrets. Those nasal samples were placed onto dog kidney cell cultures in a lab. And if the cultured cells showed CPE cytopathic effects, this was interpreted as evidence that the virus was present and replicating. And they also measured, quote, antibody levels in the ferret's blood, and a rise in antibodies was treated as confirmation of infection. So I think we see the problems with this, obviously, and this is also a perfect example of what's done with respect to animal models in order to claim transmissibility of a substance. And in many of the cases, if not, I guess you could possibly say the overwhelming majority of cases with these animal models, which I'll get into some in a little bit, those who are set in the cage next to the animal that is inoculated with an absurd amount of unpurified fluids or something like this don't get sick. They don't get sick. Invalidating transmission. Okay, so then to test whether the adapted material could cause severe disease, they injected a large volume, 3 milliliters of culture mixture directly into the ferret's tracheas. 2 ferrets died after having that large amount of fluid injected into their lungs. Again, the material was still an unpurified tissue culture mixture. No clear control experiment was described where the same volume of non-viral culture fluid was injected into ferrets to see whether the procedure alone caused harm. So this is an example of gain of function. This is what's done. It's riddled with unproven assumptions using animals that are treated horribly in a laboratory environment, pumped with an insane amount of stuff. And when they try the transmission stuff, just because they find genetic material or differences in the genetic material from one ferret to another in cages next to each other, they assume that means that the virus has mutated. And then when they dump an absurd amount of unfiltered fluid into the lungs and directly into the tracheas of the ferrets and they die, they claim that that proves that the virus was more viral. Anything to add, Jacob and Liev?
No, it's just animal abuse. I mean, virology has roots in Louis Pasteur, who notoriously abused animals in his experiments. There's a lot of other experiments done where they will combine cell cultures of different supposed viruses and then make a chimera. And then they take that and they inject a large sum of that fluid from a cell culture into the lungs or the brains or the cavities of various animals. It doesn't always have to be ferrets. It could be rats, it could be mice, it could be bunnies. And the way they have to resort to doing this is absolutely insane. They take this mush and they inject it into their eyes, into their kidneys, whatever the case is, and they just wait for something to happen. And then whatever happens, they'll say, "Oh, this is proof that there's a virus." When part of actually both Rivers and Koch's postulates was the ability to make the same disease when exposed to the virus. But in many cases, as you mentioned, these animals are actually getting different diseases or just not getting sick at all, or they just die because their bodies just get shocked. And then they use this as proof that, oh, we're building up the virus, it's evolving, and it's none of that's happening at all. They're just killing animals.
Okay. Can you do gain of function on Santa Claus? Can you do gain of function on a unicorn? Can you do gain of function on a garden gnome with lasers for eyes? No. Could you possibly do gain of function on a bacteria or fungi? Theoretically, it is possible. Bacteria and fungi have been demonstrated to exist. You cannot do gain of function on something that has never been demonstrated to exist. It's interesting, when I was on the high wire, I brought this up either in the publicly available session or the session that was just for Dell's donors. And he brought up an example of, he recently interviewed a world-renowned virologist, I think from France. And he asked him about gain of function. And this world-renowned virologist said plainly, yeah, we've tried this. We've tried to weaponize viruses and it doesn't work. We can't figure it out. Now, I'm sure some people would take that to mean, oh, he's lying. He's trying to cover for gain of function. That's what they say about us too. But again, when you look at the publicly available experiments, I'm not saying they're not trying to do something in a lab, nor am I saying that all virologists are in on this grand scheme, but it's, we see here, based on a variety of unproven assumptions, where what they think they're doing in a lab, as evident by a few molecular biologists that have messaged me. I'm sure Jacob and Liev have experienced this as well, and Cowan and others, that I legitimately thought I was working with viruses. But now that I see what you're saying, I realize that I didn't even question the foundational assumptions. Okay, continuing on. What about viral genomes? Okay, so this one, I still am learning to articulate this better. I would recommend reading Dr. Mark Bailey's paper, A Farewell to Virology, where he does an extensive breakdown on viral genomics as it pertains to SARS-CoV-2, which gives you insight into viral genomics as it pertains to pretty much any other virus. But so this is what's called the Fan-Wu paper, a new coronavirus associated with human respiratory disease in China by Fan-Wu et al. And this was published on the 3rd of February, 2020. And this was used as the reference genome, quote unquote, viral genome for SARS-CoV-2. So basically what happened was a 41-year-old guy in Wuhan presented to the hospital with pneumonia. And for whatever reason, living in a city that is known to have a ton of pollution, a ton of just overall environmental issues, let's say, they decided that this dude had uncharacteristic pneumonia. And from my understanding, they tested him for quote unquote known viral illnesses, and he tested negative. And rather than considering that, oh, this could just be pneumonia caused by a variety of other factors in the environment, we already have the presupposition that this is a virus. So they basically conducted sequencing on his bronchoalveolar lung fluid, and they extracted every fragment of RNA that they could from this unpurified sample of his lung fluid. They then converted it to DNA and sequenced it, producing what's called reads, and they had over 56 million short reads that were roughly 150 base pairs, because they had converted to DNA. If we're talking about RNA, it would be just bases. 150 base pairs long. So 56 million fragments of RNA that are 150 bases long, essentially. So these short fragments were fed into a computer assembly programs called Megahit and Trinity, which basically attempt to reconstruct longer sequences by algorithmically overlapping fragments into what's called contigs. So Megahit, which are also theoretical, by the way, Megahit produced hundreds of thousands of contigs in one long sequence, which was 30,000 bases long. And notably, it was templated against another quote unquote coronavirus genome that was assembled via the same way, by the way. And this templated genome was called BAT-SL-CoV-ZC45. And they said that this genome that the computer had algorithmically assembled was 89% similar to that other genome, which meant that it was just another coronavirus. So again, that 89% similarity was treated as meaningful, meaning they were related. But if we think about that, human beings are said to share roughly 96 to 90% of the same DNA as chimpanzees. And we are very clearly different than chimpanzees. 89% similarity, and they're saying that they're from the same, they're basically like the same viral family. And again, this is all computational algorithms assembling RNA fragments of unknown origin, never coming from purified virus particles directly from the fluids of a sick person. Just to continue on. So this is another important note. So Megahit came up with a genome that was 30,000 roughly bases long. When they uploaded the same RNA fragments into Trinity, it generated a genome that was roughly 11,700 bases. And that's relevant because if they had only used Trinity but not used Megahit, they would have come up with a genome for a virus that was completely different. I think you can guess why that's problematic. Then what was done, PCR was then used to amplify selected segments of that assembled model, describing it as confirmation. Also notably, the Corman and Drosten paper, which was published to set forth the PCR quote unquote testing protocol for the entire world, they even admitted in that paper that these primers for PCR were made without a purified virus particle. So when they came up with these quote unquote PCR tests, the primers were made based on this computer algorithmic assembly process that is ultimately coming from fluid from a sick person that, there's countless sources of genetic material inside an unpurified sample of fluid from a sick person. So we could just say these pieces of genetic material, if you will, are really of unknown origin and unknown provenance. Jacob and Leah, do you have anything to add?
No, not really. Just a lot of computer games. And none of this, again, I cannot reiterate this enough. None of this is actually real. All of this is extrapolating non-specific sequences that are made from a code from a computer. The computer spits out a bunch of possibilities, and they arbitrarily will pick the longest one and then match it up with other sequences that they got to do in the same way. None of this is actually coming from a human body. All of it is completely theoretical. And as the most important part of it being that they created an assay or a test rather, based on these findings on a computer without anything from the person that was sick. They got a generated sequence and they sent out that test to everywhere in the world and perpetuated a pandemic because they didn't know where these sequences were actually coming from. Years later, they find out that they're pretty common sequences, but obviously we find that out way after the whole virus lie. So this is why another thing, I don't know if how much we're going to talk about genomics and stuff, but this is another issue entirely about not only there's no virus proof, not only do microbes not cause disease, but the idea that genomics is even a valid science is a huge thing.
Well, I am going to touch on that just a little bit here in a second. So just to summarize, no viral genome has ever been sequenced directly from a purified sample containing only the target virus, quote unquote. Patient samples always contain a mix of human, bacterial, and other genetic material, and total RNA is extracted from all of it together. No viral genome has ever been read in one continuous physical read from start to end, not even with long read sequencing, although technically feasible, and not even with Sanger sequencing, considered the gold standard. Every published genome is stitched together from overlapping fragments. Published viral genomes are computational assemblies, not direct readouts. Software algorithms take millions of short fragments, 150 bases of mixed origin, and attempt to stitch them into a candidate sequence. The first published version of the SARS-CoV-2 genome actually contained confirmed human, quote unquote, DNA, matching the human genome, which I'll get to the problems with that in a second, at start and end, confirmed by Edward Holmes, a co-author in writing. It was quietly removed in later versions. 5, there is no independent verification loop. Every subsequent study that confirms a viral genome uses primers and alignment references derived from the original computationally assembled sequence. So when you hear about, oh, there's a SARS-CoV-2 variant circulating about, or a variant of the flu circulating about, it's using the same computational algorithmic assembly process, just getting a slight variation. Maybe it's 97% similar, 96% similar, 98, maybe 99, found in the fluids of a sick person coming from an unfiltered sample of said person's fluids. And then they say, oh, because this is 99% similar or 98% similar to this other reference genome I have, that must mean the virus has mutated a little bit. So both the US CDC and Germany have formally confirmed that they do not isolate viral genetic material from other genetic material before sequencing, and neither possess a data set where the complete genome was independently reconstructed from scratch. The bottom line is what they call a viral genome is a computer model built from the fragments of unknown origin, not a molecule that was physically observed or read end to end. And this is all coming from, by the way, I want to give a huge shout out to him, Ben, I think his name on Twitter, on X, is US Mortality. Highly recommend checking him out. He's done a lot of breakdowns on the problems with viral genomics. This brings up an important point, though. So this may have, they may have claimed up until this point, or after this, that they have fully sequenced the human genome, although I highly doubt that. But as of this article in February of 2023, so 3 years ago, which is titled "Why the Human Genome Was Never Completed," they describe how the human genome has never been fully sequenced end to end. I can see a human being. I can look at a human being. I can isolate a human being. Maybe not when you start learning about ether and stuff like this, but the point is I can isolate a human being. I can see one. I can interact with one. And they still have not mapped the human genome. So you expect me to believe that they know the exact genome with the exact variations, the exact parts that are problematic, the exact parts to test for a particle that they have never demonstrated exists in the first place, by their own admission, by the way, with Christine's FOIA attempts and things like this. I think that's absolutely insane. And there's a whole separate category of things we could get into, whole separate discussion on the problems with the genetic, the dogma of genetics, or the DNA dogma. I forget what it's called, by Watson and Crick, which was what established the double helix model of DNA. Okay, before we move on, anything to add? Liev, and how are you doing on time, man? Because I know we got to get to yours sometime soon.
I'm good. Yeah. Good for another 45 or so.
Okay.
One of the big things with the human genome too is allegedly there are viral genome in our genome. They can somehow make that claim. Again, in the kind of the convoluted scene, we fill our heads with so much information in genomics. I mean, I studied genomics primarily in my undergraduate degree. I was in lab. I've done the labs on allegedly isolating DNA. There's a huge disconnection between, well, there's like layers to it. There's real life, then there's lab science in a test tube, and then genomics and genetics takes it a step further and we go into the computer, right? Like we're so disconnected from reality, from anything that we can actually study empirically. And that's one of the biggest problems. We fill up the textbooks. I mean, the textbooks on genetics, they're huge, full of assumptions, right? There's no scientific method employed. In my entire undergraduate degree, we never went over the foundational papers. Like Watson, we didn't even read his paper. We didn't read any papers on genomics. We didn't apply the scientific method once. In the laboratories, it was all just typing on computers. And I mean, this is why people, they get convinced just because we make so many claims. We make so many claims about genomics. Oh, it's genetic. Oh, it's genetic. This is this. And we trick people into thinking that we have all these applied sciences. But that again is kind of just a farce too, right? Like we point at genomics when it comes to GMO, but it's actually all the harm is coming from the toxicities, right? Like we just try to point always to genetics, even though we're looking at confounding variables that are actually creating effects in the world. And that's sort of the difference. It's going back to the philosophy of science and empiricism, and that there is no scientific method. A lot of these claims are unfalsifiable, which make them pseudoscientific right out the gate. We engage in circular reasoning, theoretical backfill, theory latedness. It's just a whole convoluted batch of nonsense.
Well said. Okay. So I'm not going to get into PCR. Basically, PCR, quote unquote, tests are the primers for it were created based on this reference genome, and it's ultimately RNA genetic material, if you will, of unknown provenance, unknown origin. So when we talk about PCR, I did interview Yernea Tomsik, who has worked with PCR for quite some time. She's a molecular biologist, and she highlights all the same issues that the three of us are sharing here and shares the same perspective on that. But when we talk about PCR, we're not talking about false positives. We're talking about just false results, period, because there is no virus particle. And again, want to share both The Final Pandemic by Doctors Mark and Sam Bailey with respect to all that we've covered so far, and then virology.com, especially when it comes to specific quote unquote diseases, and then also electron micrograph images. Okay. So I'll cover logical fallacies after we get into bacteria, fungi, and parasites. I'm going to go ahead and turn it over to Liev first. I'm going to stop sharing my screen and turn it over to Liev for him to share about parasites. So let me go ahead and spotlight him for everyone. Take it away, my man.
Thank you. My primary claim about parasites is that the concept does not exist in nature, right? And that's a bold claim, so we'll get there. But that's where I want to start off, because the concept of a parasite, the idea that somehow in nature we have take without give, is again a reduction of our perspective, right? It's when we dive too deep into the lab science, as we've seen so far in this presentation, we lose the holism, the whole of nature, right? And so when we look in the gut and we see a parasite, right, we instantly go to that, oh, that must be the cause of the discomfort, of the disease. Interestingly enough, parasites have the highest level of asymptomatic rate, right? Like billions of people have worms, and yet the symptomatic rate are very low. So we run into a problem right away where many people harbor these so-called parasites, yet they don't have symptoms. So again, we have to get back to the basics here, right? Like obviously right out the gate, we understand Koch's postulates are an impossibility, and this is admitted in parasitology. You know, even esteemed parasitologists admit that Koch's postulates simply cannot be conducted with parasites because of the high asymptomatic rate. So when we think about this, you know, isolating the organism from the disease state, it's like what causes disease if it's not a parasite necessarily? And why are parasites sometimes present at the site of disease? Well, the answer lies obviously in the terrain, as I think we can all kind of assume at this point. And underneath the parasite, what we see is most generally, from a physical standpoint, toxicity. Right? That is truly at the root of, in my opinion, and my read of the literature at this point, at the root of all so-called infectious disease is most generally toxicity. Because these organisms, and Jacob's going to give us the rundown on fungi and bacteria as well, the role is the same for parasites. They're present at the site of disease, not to step on Jacob's toes, but I know he's got lots of wisdom for you guys. The role of all of these organisms truly is to heal the environment, right? It's to bioaccumulate toxicity and bioremediate toxicity. Those are the primary roles of parasites, as well as these biological organisms. individuals who cleanse constantly. Right? This is kind of another permitted narrative. Oh, take ivermectin. Everything's caused by parasites. Cancer is caused by parasites. If a parasite is in a tumor, there's toxicity. And people who constantly cleanse, huge studies, 84,000 people. There was a trial in India where they gave parasite cleansers to children, 2 million people in this trial. No reduction in any negative symptom. Like all-cause mortality didn't go down, height, growth, like IQ. They tested all of these things, mental cognition. None of these things were actually lowered. There's actually a ton of studies that show that parasite cleansing can lead to increases in ulcerative colitis. They can lead to things like allergies, asthma, eczema, autoimmune conditions, gut disturbances. And there's actually an emerging field of helminth therapy. People are with IBD and they're taking helminths willingly and their symptoms are gone, vanished. They can consume foods that they couldn't consume for years, right? They always blamed, oh, it's the gluten. But they take helminths, they re-expose their bodies to these worms that are, again, innate part of our existence. And at the root, their symptoms are going away. It's amazing. So there's a ton of literature now on the connection of helminths and how they regulate the so-called immune system. I don't love that terminology, but if we think of the immune cells, right, there's a very great connection in how these organisms like parasites can interplay with these internal systems. I would argue that we're not separate from the microbial world or parasites. As Jacob's going to point out, he's going to talk about pleomorphism. These are innate parts of our bodies, even though the parasite life cycle is seemingly exogenous, that it comes from with like outside, it's still a part of us. Like we're not separate from nature. And as we've sterilized our entire world, health outcomes are lowered. So there's a lot of empirical evidence on this, and you can approach this discussion in many different ways. You can approach it logically, where of course the asymptomatic concept completely contradicts, like 100% contradicts the concept of parasitology. Koch's postulates cannot be attained. We can ask questions about the presence of these parasites in the first place, helminths, everything. And even the most esteemed microscopists admit that many of these parasites are elusive under the microscope. And so most of the parasitology literature actually relies on PCR and serology, right? Antibodies, which are non-specific. So we've covered in virology very clearly how a lot of these laboratory methods are extremely problematic. And even in the parasite world, seemingly these are bigger organisms, but still they're elusive. Parts of their life cycles are elusive. They can only be seen when we stain and alter the sample. So there's all of these questions in the lab setting when it comes to parasites, which it's very important to consider this, because the virology starting point is so important. And then we begin to extrapolate it to the other fields. Like a lot of this stuff is much more simple than we think. We say there's trillions of different species of bacteria. I think that we're on the same page that that's only perpetuated by genomics.
Correct.
Right? When you look under the microscope, there's not trillions of bacteria. There are distinct forms that you can see. And these forms, I believe, certain forms have certain functions, but there's not trillions of different species. That's all perpetuated by genomics, which have the same inherent problems when it comes to parasites. Right? And so to leave this note of parasitology, I mean, we could dive into protozoa, right? The single unicellular parasites like Giardia, Blastocystis. And there have been trials. There have been human challenge experiments where people have ingested protozoa, they have ingested helminths like worms, and they don't get disease. People have tried to take, there was this one guy, Grassi, he's most notably known for his malaria papers, and he tried to give people malaria through mosquitoes. Never ever was able to produce any results. So he himself actually has ingested multiple different parasites over the course of his lifetime. I found literature on 4 or 5 different types of parasites that he ingested willfully, never had diseased once, never got diseased once, even though he ingested all of these different types of parasites. And there are accounts in the literature that I couldn't find references for directly, that he consumed more. So this guy Grassi was just out. He was an Italian guy, and he was just consuming parasites, and none of them were causing disease. I found about 75 papers across the parasitology literature. It's not as vast as the rest of the germ theory literature of people doing human challenge studies, but about 75 papers of people consuming protozoa or helminths. And the only time that they can produce disease in people by giving them parasites is if alongside the parasite, they give a toxicity, right? So sometimes they store parasite eggs in formalin, right? A formaldehyde derivative. And then they give it to people, and the people get a little sick tummy, and they're like, bang, parasite causes disease. It causes GI upset. It's like, okay, was it the formalin or the parasite, right? We have to isolate the variable. And this is not what's done in any of the germ theory. This is done across the board. The only other time that I found people had symptoms when they were willfully ingesting parasites of all kinds was when they cleansed prior to ingesting the parasite. When you cleanse in a forceful manner, right? You try to kill certain things or force certain things out of the body, you disrupt the microbiome. When you disrupt the microbiome, the protozoa can come in, or the parasites in general can come in, and they do cause symptoms sometimes, not all the time, but still the prerequisite is a cleanse. Now, in the literature, they've shown more severe cleanses. They've actually used to do a lot of these, new age cleansing back in the day, right? Like taking certain pine derivatives or castor oil. Like they used to do more natural things as cleansing, and still, if you ingest a parasite after the cleanse, you can develop symptoms. So the important part of this discussion and all of the exploration of germ theory really is looking at the logic of when is there actually disease? Because if the organism is present and there is no disease produced, how can we confidently say that the organism caused disease? At the root, there are terrain factors. There's the underlying terrain. And I would posit that all biological, what we call parasites, all have a positive beneficial role in the body. And so parasite is a misnomer because of their bioaccumulation, their bioremediation. And so I'll touch on briefly, the most exciting part of my work was exploring ectoparasites, things like lice, fleas, mites, ticks. And what's really interesting here is when you study paleopathology and anthropology, which is essentially the study of disease in primitive people, pre-civilized people, the occurrence of ticks, mosquito bites, lice, tungiasis, flies embedding in people's, it's all non-existent. So even the concept that now we're constantly inundated by bugs and ticks, primitive people were actually bit by bugs less across the board, even mosquitoes, which aren't considered ectoparasites because they don't stay on the host, right? So that's a really interesting observation that I found. That's still there's really a lack of literature in this area. And so I rely a lot on sort of the foundations of the terrain and logic to make sense of this. I don't really have the empiricism to back it up fully, except for the anthropological literature and the paleopathology, that primitive people who were in pristine health were not affected by these ectoparasites. So the other side of it is that leeches or maggots are used medicinally sometimes, right? So that even the presence of a mosquito coming and taking a little bit of your blood might just have a positive effect. And in my fundamental understanding of life in general is that everything happens for a reason. All is extremely interconnected. And that everything has a purpose. And operating on that is my baseline. I'll admit, every single time an insect bites you, there's a reason for it. My empirical evidence from this, I do extrapolate from Thomas Dijkstra's work, Dr. Thomas Dijkstra. And he works with plants and insects. And he says that healthy plants aren't affected by insects. Insects don't eat healthy plants. And he measures this in Brick scores and the amount of sugar in a plant. Right? So the same concept that these healthy humans aren't affected by insects. Right? It's like the same concept. And so when we're in the optimal terrain, when the plant is growing in the optimal terrain, the insects don't bother it. And so these organisms, they all play this bioremediation role, this adaptive role. Thomas Dijkstra is his name. Dr. Thomas Dijkstra showed this. His entire life work is centered around this. Dykstra. Dijkstra. So the primary role is always going to be some sort of greater adaptation. And so my ask when we look at parasitology and nature in general is that we must consider the whole. The whole is always greater than the sum of its parts. And when we hone too close into the parasite, we miss the ecology, we miss the terrain. Right? So that is at the root, at the foundation, the entire point that I believe many of us are trying to make here. I mean, all of us here in this discussion, we have to zoom out. The lab science, there may be an application, right? There might be an application for empiricism. I think we have to be careful. I think we have to get back to Popperian falsification. I think that's the only reasonable way to apply empiricism. And we must be careful of positive claims. But we have to zoom out, right? When we look at these diseases allegedly caused by parasites, we have to look at what what are the symptoms? Because someone can say, oh, well, I have Lyme disease. But what is Lyme? Right? It's an entire plethora of symptoms. Like dozens and dozens of different symptoms can be Lyme disease, apparently. Because all Lyme is an antibody test. So they give anyone who has symptoms who they're like, oh, I don't really know what happened. Do you ever get bit by a tick? Yeah, like 3 years ago, I got bit by a tick. Oh, I have this Lyme test. Oh, it's Lyme. And we've created the disease, right? The symptoms are totally non-specific when it comes to Lyme, when it comes to malaria, when it comes to any of these things. And they're all new age diseases. Study anthropology. You realize that there was no malaria before there was the modern world, before there was civilization. Before we altered the terrain conditions, there was absolutely no disease associated with parasites. You know, maybe there was some cyclical adaptation to the world around us. I'm not going to necessarily deny that. But there was no chronic disease and there was no infectious disease of any kind. No primary amoebic meningitis. There was no Lyme. There was no malaria. There was no little GI upset from drinking the stream water. That doesn't happen. Primitive people consume the most stagnant water that you can ever imagine. They dig a little hole in the ground, they can drink it because their bodies are adapted to nature. We are disconnected from nature. We are sterile. And so my non-medical advice is go and have a drink of a stream that's pristine. Sometimes I go and do that to rewild my gut, to rewild my connection with the earth, the soil, with nature. Right. And in that, I developed that resilience, that strong baseline. That's the point. It's the rewilding. The concept of parasite, the idea that there's an organism that has no greater role than to just cause havoc and harm to people is absolute nonsense. And that is a lie that we have projected onto nature because we as humans have been corrupted. And there are humans out there that cause havoc on other people for seemingly no reason. But I would still say that there are deeper connections involved there that we can explore spiritually. There's always a reason for everything. So I'll leave it there. Jake, do you have anything you want to add to that, buddy? Or
No, it's like a perfect segue to what you were saying. At the end of the day, as the Bigglesons say, there is no antagonism in nature. There's only symbiosis.
exactly.
Everything is working with a reason. We as humans, since we like to divide ourselves from nature, want to be like, no, we're not part of that symbiotic process. We're special. We're separate from all that, when that is not the case at all. Alec, you want to say anything?
No, I just wanted to give Liev the opportunity to answer some questions since I know he has to go. Is that okay with you?
Sure. Yeah,
Okay.
yeah. Let's do it.
So the first one, let's see. I'm trying to find just the parasite specific ones. For Liev, I've been thinking that parasites aren't the problem, but are the firemen. My question is, where/how do worms show up in intestines, for instance? Are they larger pleomorphic forms of bacteria?
Oh, this is
Yeah, this is a
That's
good
good.
question. And Antoine Béchamp actually did answer this in one of his works. I do agree with Antoine Béchamp's point of view here. Based on the literature, we can be exposed to these things exogenously. They can come from outside, right? Because it is true that you can see colonization of the body of these organisms when we ingest them, like their eggs or their larvae, especially when it comes to worms. So it is possible to get them from the outside. That being said, the terrain is still everything. You know, if Jacob, Alec, and I all consume parasites, maybe it takes hold of one of our bodies who needs it at the moment. Maybe it doesn't in two of us, maybe all three of us, because we tend our terrains. We'll be fine. Right. So it always comes back to the terrain. But yes, they can come from exogenous sources. But again, we are not separate from nature. Right? Like, we are not separate from the worm in the soil, right? Or the parasite in the soil. Like, we're supposed to be in direct contact with the soil. Our food should have some soil on it, should have some of these eggs and these larvae. Being around animals like dogs and cats, you know, they allegedly harbor these parasites. Like, this is not the harm. We're not disconnected from nature. It's actually in the deworming that we disconnect from nature, which then sometimes that's why we get a hookworm, right? It's like nature's re-entry to our body, you know. But I know Weston Rowe, he had a hookworm that came up through his leg. You could see it came through his foot. Totally fine. He let it pass. He had a little rash. It came and it went. Nothing came of it, right? It's only when we go and take the parasite cleanse, the ivermectin, the fenbendazole, and then we get in our own way. And that's where the true problem lies. Like, we're not separate from nature. And the concept of pleomorphism is so interesting too, because we have that internal pleomorphic mechanism that Jacob's going to mention here. And the parasite, it takes a very similar structure in development, right? And someone once said to me, oh, a life cycle of a parasite isn't the same as pleomorphism. I'm like, okay, but if you look at them under the microscope, I mean, you see very clearly that it all looks the same. Like, it's the same fundamental process, just maybe because hookworms don't come from microzyma or bacteria, doesn't mean that we're any more disconnected from this pleomorphic nature of these parasites, because you can ingest the eggs. Sometimes they develop, sometimes they don't. It's all based on the terrain.
Real quick, for those who don't know what microzyma or pleomorphism or any of that is,
Jacob's going to cover it.
Cover that.
I got it. No worries.
Jacob's going to cover
And one
it.
quick thing on the colonization piece, Liev, as I'll share, and some of the animal models that supposedly